this talk. Okay, so it's my pleasure today to introduce today's speaker, Ashley Bordelon. Ashley grew up in the creeks and forests around the Dallas-Fort Worth area, but she truly discovered her passion for the environment when she was at the University of Alabama getting her biology degree. So during her time in Tuscaloosa, that's not woo pig suey. What are you guys are something else? Oh, we could talk about that in a sec. But during her time there, she worked at the University of Alabama Herbarium, focusing on imaging specimens. And then in 2017, Ashley was here at BRIT working as a summer intern, and that was during the time when we acquired the Louisiana Monroe collection, and which is like 470,000 specimens. So she had the joy of working on that over the summer. And then in 2019, she came back and was responsible for managing herbarium volunteers, doing other activities related to curation, working on staff at BRIT. And then in 2020, Ashley started working remotely as a digitization technician, and it's really just, now she's full-time. A lot of you know her because you work with her as volunteers. And she's just really truly fabulous, and we've discovered her as an herbarium. We consider her an herbarium star these days. So without further ado, I present to you today Ashley Bordelon.
Thanks, Brooke. I'm flattered. Like Brooke said, I'm currently in Lubbock, Texas, way out west. I'm interested in a lot of plant groups, even though right now I'm working exclusively with specimens from Texas and Oklahoma. I really have been enjoying fungi and myxomycetes over the past few years. It's been really fun to learn, and I've learned a lot from BRIT researcher Harold Keller, who's kindly joined me today to help me out with any questions you may have at the end. So Harold and I are just two of the research team that are interested in discovering smaller life forms that can be found on live trees in urban environments. So this map that you're seeing right here, we're located in north central Texas. We're in Tarrant County. So we have this Tarrant County map with an inset map zoomed into the Fort Worth Botanic Garden and BRIT campus. So over the past few years, we've discovered new and interesting fungi, myxomycetes, and we've even discovered a microscopic beetle on our own campus and in local nature parks. So if you're looking at this map, you can kind of see, it's a very urban environment. There's a lot of roads, a lot of buildings and impervious areas, with just a few pockets of, this greener area, these parks and local nature parks. So our focus hasn't necessarily been on going to these larger nature parks, but just in general, we're looking for large, older trees in urban environments. So places like neighborhoods and along the streets as well. We even discovered one group of white crust fungi had a higher species diversity along streets in a neighborhood than those found in a nature park. So that was really interesting. Most of our collections and discoveries are made outside in the field, but some have been made, after careful observation of bringing these collections back and watching them over days, weeks, and years back at the BRIT building or in the Sumner laboratory. So these fungi and myxomycetes in particular are what we're going to be sharing with you today with the moist chamber techniques that we use to observe them. I want to mention amateurs to start off because I definitely feel like I am one. I'm learning a lot over the years, so I don't know everything, but it's not a bad thing. I think amateurs are really important in any fields. I think they bring a lot of continuous curiosity and passion, especially in the fields of mycology and myxomycology. Many new species have been described by amateurs. The British Mycological Society was founded by groups of naturalists, both, professionals and amateurs. And if you're familiar with Beatrix Potter, the author and illustrator of the Peter Rabbit books, she also had an interest in botany. She wrote a paper on spore germination, which was really interesting. There's this quote right here. Beatrix Potter was nevertheless a pioneering mycologist, one whose intelligence and inquisitiveness might have been channeled into a career in science had she possessed the Y chromosome required for her required for most Victorian professions. Fortunately, her considerable artistic talents gave her other outlets for her ambition. Amateurs in these fields today are largely important because there's a very small number of scientists that are devoted to these certain fields compared to others. We know that these organisms are really important, but there's not large public interest and therefore less funding for these groups. Let's see. An interesting article I read recently actually said, even current mycologists, there's a lack of unified community among them in the fields of biology. So our group in particular, I think at Brit has been really special because most of us are amateurs with a few exceptions, but each one of us has an ability that we can bring to the table and collaborate with. So before we jump into it, I'm just going to give you a brief basic information about these organisms, fungi and myxomycetes, and then we'll go over those moist chamber techniques. So what you're looking at right now is a tree of life. For those of you who aren't familiar with just how old some of these organisms are, this website, Evo Geniea, was really interesting. It's an interactive tree of life, so you can click on some of these. But bacteria and archaea are our oldest, and they're all the way on the far left, and then followed by all of the eukaryotes, which includes you, me, and the fungi and myxomycetes. However, we diverged from these organisms billions of years ago, but they are older than us. So with all the recent rains recently, I'm sure that some of you guys have been seeing fruiting bodies popping up in your yards, mushrooms. Those fleshy and capped mushrooms are typically what people think of when they think of mushrooms, but there's a lot of variety in them. There's a lot of size and structure differences in the habitats that they like. But basically overall, they're in charge of recycling dead and decaying material. You know, they're the trash burners in nature, so our landscape doesn't get cluttered up with dead material, dead plants, and they're really ecologically important for recycling carbon. Universities and institutions like ours store specimens of fungi in scientific collections. So what you're seeing right here is a specimen from our Vanderbilt collection of Amanita roanoke incense. So typically these mushrooms are stored in archival boxes and either have some kind of label inside or on the outside of the box. If you can read it from your screen, the label in the top left-hand corner has in parentheses with odor. So notes like these from the mycologist who collected this made some useful notes that could potentially help identify it later down the road. So this species of Amanita in particular has a smell of like meat, or it can smell like carrion. So it's very distinguishable. And if Amanita sounds familiar, the popular cousin of this species is that white spotted red mushroom, Amanita muscaria, which is known for psychedelic effects. So that might sound familiar, but not all of them are psychedelic or poisonous. Morel mushrooms, for instance, you may be familiar with. These are delicacies, the Morchella species. So you can find these again with American elm trees. And the recent Fungi magazine issue features our own Harold Keller's article about morel mushrooms. So you'll have to check that out when it becomes available online. And then this example is a not so obvious fungus. The vertical white splotches that you see on this tree are a white crust fungus called Dendrothele nervosa. So this species in particular grows specifically on this eastern red cedar. So we found it a pretty interesting group to study recently over the years. Fungi have different ways of living. They can be saprophytic, they can feed on dead organic substances, they can be parasitic, they can absorb nutrients from live plant or animal hosts, or they can have symbiotic relationships with other species in which both benefit. So this Dendrothele is usually found on large live trees rather than all the dead branches or dead trees. So we're not sure what that relationship between the tree and this species is right now, but we're looking into it. And then now on to our slime molds, our Myxomycetes. We can also call them our little slimy friends. They can be called biological jewels of nature. They're very different. They've been lumped into the kingdom Protista with a bunch of different other weird and unique organisms. Some of those are algae, diatoms, and amoebas. So just a weird group because they don't exactly fit with the plant kingdom or the animals, but they do act more so like animals than plants. So their role in the ecosystem is even different than fungi. So rather than feeding on organic material, they hunt and feed mostly on bacteria. So they're predators, but even other microorganisms and fungal spores as well. So this is a good quote from Thomas McBride. Slime molds may occur on objects of any and every sort. Their minuteness retires them from ordinary ken, but such is the extreme beauty of their microscopic structure, life, the exceeding interest, such the exceeding interest of their life history that for many years enthusiastic students have found the group one of peculiar fascination, in some respects at least, the most interesting and remarkable that falls beneath our lenses. And I've grown to definitely agree with this quote. The picture you're seeing on the right is a species called Lamproderma achinulatum. A naturalist in Tasmania, Sarah Lloyd Ohm, took this picture. She has photographed tons of slime molds in Tasmania. She's contributed over 1,000, almost 2,000 collections of Myxomycetes, representing about 120 species. And all of these species were found within 2 kilometers of her home. So pretty amazing. She didn't have to go very far to run into all of these species. And she's what you would call an amateur for sure. If you've never witnessed what a slime mold can do and a slime mold on the move, I'm going to show you guys a video. Let's see. So this video shows a slime mold starting in this center, followed by, you see all these pieces of food spread out the area right there. These pieces of food are meant to mimic cities in Tokyo. So as the Myxomycetes starts going after the food, it begins to leave chemical cookie trails along the way, eventually making the strongest connections with the shortest possible route. So after this experiment, they realized that this map made by this slime mold ended up looking a lot like the railway system in Tokyo, which is pretty cool. This is the structure you can see on the right-hand side. This species is Macbradiola denticulata. So it's definitely different than the fungi and the way it moves and all of that. So let's go to the next. Here, I had to show a specimen also of a slime mold. So this one is Trichia persimilis. So the fungi that we saw was in a box, but sometimes they can be in bags as well. However, the slime molds, the Myxomycetes, they need to be stored in some kind of 2-part container. So typically you'll see the substrate, whether that be the piece of wood, the leaf is going to be glued on the top of this box. And then the label information, that data is going to be on the other side. That way the data and the specimen are never separated and we know what it is. So we've gone over these different life forms. Now we're going to go into exactly how to make the moist chamber cultures. So the main ingredient of these moist chamber cultures is the live tree bark. So there are a few species that have shown to be really favorable for these organisms. That being these 2 right here, the eastern red cedar, Juniperus virginiana, in that photo on the left with Harold standing in front. And then we have a picture of the American elm tree, Ulmus americana, on the right-hand side, showing that diagnostic trunk structure. While both fungi and myxomycetes can grow in all types of habitats, the species we're looking for specifically have chosen these tree bark species as their niche. So these pictures show the tree associate, but not necessarily the habitat that these organisms are in. So fungi and myxomycetes can be so small that we need to look at the world from their view by looking closely at the bark rather than a step back looking at the entire tree. So what makes the bark of these tree species so special? And what's an ideal microenvironment for fungi and myxomycetes? So, you know, Junipers virginiana and American elm tree of Ulmus americana both have bark that's not too alkaline, not too acidic, has the ability to absorb a lot of water and retain that water. They also have a lot of ridges and grooves. It's quite a landscape on that scale. And there's also a lot of the other organisms, microorganisms present with the bark as well. So both of these species have this quality. And so that's why they hold the greatest myxomycete diversity on their bark. So how do we sample this tree bark and create a moist chamber culture? I've listed the materials here. For collection, you'll need a field notebook to store your notes and your data, paper bags to hold the bark, extraction tools, so a flat head screwdriver or pocket knife, a tape measure to note the diameter of a tree that you're sampling, and then a camera or your phone for pictures of your samples. Moist chamber cultures, the technique is not new. They've been used by scientists to isolate many different organisms since the early 1900s. Traditionally, they have been done on small agar plates in a lab. However, we wanted to present an option that isn't exclusive to those that have access to a lab, but a method that has been tested and used by our own researchers and has been proven to be successful with these simple everyday household items. So we have a pie tin, distilled water, paper towels, and saran wrap. So the first step is collecting the live tree bark. But before embarking on any tree collecting venture, you do need to have permissions. But if the tree is in your own backyard, that makes it a little bit more convenient. The eastern red cedar is probably the easiest bark to sample because it's so thin and shreddy. You could even use your hands. But for American elm, for instance, you would probably need to use some kind of tool that you're comfortable with. So I have a video showing you how to do this. Removing the bark requires careful consideration. And stopping my video. Because we don't want to harm this tree. We want to make sure that we leave it unscathed and not vulnerable to attack or infection. We only need the outside layer of the bark for these moist chamber cultures. And you want to collect enough pieces of bark to fill the bottom area of your pie tin. And while you're out there collecting, you'll want to record the scientific data that's important down the road. So the date, the location, the tree species, estimated height and DBH, where on the tree was the sample taken. Maybe you want to include the cardinal direction of the side that that tree is facing that you took a sample from, and any description of the bark or anything else that you think is noteworthy for your sample. And let's see. So we have our pie tin, we have our samples. So we will start off by lining the bottom of the pie tin with a paper towel, and then we'll fill up the bottom of the pie tin with our samples. Enough to fill the tin, but we don't want these to overlap. We want to give them a little bit of space between each other. We'll slowly pour the edges with water because we don't necessarily want to pour directly on top of these samples the paper towel is going to start soaking in this water. Typically, I will use maybe 50 milliliters at first, and then if I still see that the towel is a little bit dry, I may add 25 milliliters at a time. Too much water, if we have too much water and it's pooling at the bottom, it could interfere with the development of certain species. So carefully, we'll have to remove this excess water, especially on the first day. Finally, after the samples are in there, they've been wetted, we'll cover it with saran wrap. And that creates the chamber. So right here, you're looking at the final moist chamber culture, what it should look like. I've actually appended my data to a piece of tape to distinguish the sample and to give you guys a good example. However, I put this on a piece of tape, but typically we'll put it just on the saran wrap because we want to be able to look in at the bark from the outside. The more that we view this bark, either by lifting up the saran wrap, maybe we're letting airflow in there, it could contaminate our moist chamber. So putting the data directly on the saran wrap is probably ideal. At this point, we've created this chamber, and it's a microenvironment that's going to be a lot different from the humidity and temperature from the room that it's sitting in. So you can already tell from this picture that the bark is darker in color. It's already soaking up that water that's at the bottom. This moist chamber culture should be placed in a spot away from direct light, and make sure that the room it's sitting in should be about average 68 degrees. So it's going to sit like this for the next couple of weeks, and over time you'll start seeing new growth of various different species. So you'll first observe after the first 24 hours and continue observing every 12 to 24 hours when time allows. So there aren't too many eastern red cedars out here in West Texas. The video I showed earlier was actually Juniperus monosperma. I didn't think it was an eastern red cedar at the time because I'm a little bit new to some of these West Texas species. But thankfully, Harold offered me some of his bark samples of eastern red cedar just for this presentation so I could show you guys the progression over a week or so. So I just have a basic stereo microscope with a camera that I hooked up to my computer to start taking these photographs. After the first 24 hours, I started to get a closer look at some of the existing dendrothelium, saw some green lichen on there, but a closer look, I started seeing these orange spots on here. Those are mycobacteria. And I'm also starting to see just the beginning stages of growth of Myxomycetes. Potentially in these moist chamber cultures, you can also see nematodes and tardigrades. After 24 hours, I have new growth as well. So there are a few species that start to pop up first, Echinostelia and Lycia. So the Echinostelia are stalked, the Lycia are more sessile, but those two are known to being some of the first to pop up over the first few days. 3 to 4 days after wetting those samples, I saw an abundance of this species called Comatrica. This is all on a single small piece of bark, but I was able to see the stages of growth from the very beginning. So those white individuals on the right are in the early stages of development. And then on the left, we have, they'll eventually grow into those black individuals, and then matured species are these orangish ones right here. So just this past Saturday, I observed one piece of bark in the moist chamber culture and found another species of Comatrica. Looks a little bit different than the one I saw the previous days. This picture up here showing a lot of individuals actually has, maybe 4 species just growing in this small space. So these Myxomycetes, they are millimeters in height. They are visible with the naked eye. However, you definitely can get a closer look with a microscope. I will say you don't need a microscope. I have a few hand lenses. So if you're interested in doing this, you don't need a microscope necessarily. It definitely helps. But these are stuff that you can see with the naked eye as well. And then I started seeing this leafy green structure, and then possibly a mite over here. So just less than a week after wetting these specimens, I saw quite, a diverse landscape of different species. So through this moist chamber technique, our team has published new species of fungi and myxomycetes. So on the left-hand side, we have a picture of Mycena olmi. This one in particular was found on live American elm tree bark in the Fort Worth Botanic Garden. In the center is a photo of a myxomycete called Lycia iridescens. This one was found on the bark of an American elm tree as well, from Oliver Nature Park. You can also see in this picture, there's some crystal-like structures underneath that organism, and we're currently not sure exactly what this is, but we've been seeing these crystal structures in a lot of cultures as well, and on the surfaces of these pieces of bark. Finally, on the right is a picture of spores that are really spiny of a new Lycia species that should be published possibly later this year. And also found locally, which is pretty amazing, that just within Tarrant County, on these new pieces of bark, we've been able to discover these species. So I want to thank the research team, the Fungi Myxomycetes and Trees research team, and those who have also helped and assisted with some of our projects and collections. I have some references right here. And if you're interested on our website, you can check us out, and we're gonna be posting a full-length video, not a full-length video, but maybe a 5-minute video within the going step by step through these moist chamber culture techniques, and we're gonna have an accompanying PDF with those materials and instructions. This is not exclusive to anybody. I would encourage anybody of all ages to join us in looking at these pieces of bark. And Brooke, that's all I have. And Harold and I can answer any questions.
Thank you, Ashley. That was fantastic. Let's see if you will go ahead and stop sharing. We'll get back to viewer mode here. Well done. It's so fascinating. So we have a good sized group, but I think what we can do, since we've got enough time to do it, we can, if you're interested in asking a question, you can use the raise your hand feature, and then I will kind of click over to your box and I'll ask you to unmute, and you can unmute yourself that way. So I believe Tom Green had a question. Let me see if I can find him and unmute him. Or the Tom Green user, yes. So Tom, let's see, I can't unmute you yourself, but I can just ask. There you go. Okay. Tom, did you have a question?
Yes. Can you hear me?
Yes, we can.
Okay. I was wondering what magnification was used to view the slime molds, and are there any other tree bark species that you want to next hunt for slime molds?
Good questions. I'm actually not too sure exactly what magnification I took those pictures at. I got this microscope on Amazon, but I think it goes up to 1,000 times. So I'm sure that I was zoomed in pretty far, but I have been able to use my hand lens. Like I said, different hand lenses, like I can show these videos. So yes, it's good to have higher magnification, but you can still view these with just simple hand lenses. And then the tree species, yes, I think currently the team has been focused on Dendrothele, that white crust fungus. And that has been showing up on a lot more tree species than we initially thought. We focused originally on elm trees, either Ulmus americana or Ulmus crassifolia. But we've been finding them on different oak species. Harold, do you want to chime in? Or do you want to say any other tree species that you've done moist chamber cultures with that have been successful besides Eastern red cedar and American elm?
You hear me? Hello? I would suggest you use your own creativity. There are other trees that are very productive, but there are a lot of trees we don't know anything about. We haven't tested, like Quercus stellata, which is a native tree species in the area. So just use your own imagination, creativity, and go out and collect bark, but record the species of tree if you can. And I tell you, you may come up with a new species. You may come up with a new record. So get out there, do the collecting, and if you can, share your collection with us.
And I'll just note, I'll just add on that one of the last species that I mentioned, the Lycia iridescens, that was found with the help of a student summer intern a few summers ago. So she assisted with finding that new species that I'm not sure she was expecting, but it's, you know, that's why it's such a small field that people have been focusing on. So there's really a lot that we could discover the more people that actually look.
Thanks, y'all. We've got a couple of questions in the chat, and a couple of people have their hands raised. So let me do one from the chat, and we'll kind of bounce back and forth. So, Peter, you had a question. Do you want me to read it, or you want to go ahead and ask?
I'll just go ahead and ask. Thanks, if you can hear me.
Mm-hmm.
I was just wondering about sterile technique for the moist chamber culture. I don't remember you saying sterile materials, and I'm wondering how that might interfere, like if just regular ambient mold can start growing in there, even though you got it covered by saran wrap, etc. I was thinking about that mycobacteria. That's pretty cool, but did that originate from on the bark, or did it just, is it a contaminant? Thanks.
That's a good question. Yes, they can get contaminated with certain molds. I think, Harold, you might correct me, but I think you said it's typically like a green fuzzy mold that you might see, and that would be more of a telltale that it's been contaminated rather than it originated from the bark. Because the saran wrap doesn't completely, I wouldn't say it completely seals it all the way tight, but we did not use sterile techniques. But I'm sure that, you know, in other ways, maybe using the agar plates, maybe they use sterile techniques. Harold, do you want to comment on that?
Well, one way you can control how much outside aerial contamination occurs is to not spend too much time with the saran wrap clear off looking at it, although I'm sure you'll want to spend some time. And if you get a contaminant, you can take tweezers or a forceps and take that piece of bark out, throw it away, and you can control how much contamination gets into your plate. But the longer you keep the plate, the longer you take to look at it, the greater chance of contamination. And by the way, we hold these plates up to about 4 weeks because there is a succession of different organisms. Some come up quickly, maybe in 24 hours. Some come up in 2 or 3 days. Some come up in weeks. And some are really slow, and they may come up in 3 or 4 weeks. So that's kind of an overview of what you might expect to see.
And I'll say that, whenever I was taking these pictures, I would, there's no tape on that, on our, pie tin right now. So really I can just view in and you can see from the naked eye, tiny, tiny specks. Either they're going to be white, black, some orange. I could barely see that mycobacteria. And I think Harold said that's probably one of the ones that you'll see popping up first. So it's not a contaminant necessarily. But once I find the piece of bark that may have something new, I'll just take that piece of bark out only and make sure my saran wrap is still on the tin while I'm working with it. So definitely taking it off as little as possible is best.
Let me say also, you can make a real contribution to what we're trying to do, because I was surprised to find all these new species. We've got 2 more, and we will probably find some more. So what you're doing here and observing may help us a lot with our research. So get out there, get out in nature, collect the tree bark, set up your specimens, and contact us. We look forward to working with all of you.
Great advice. Is that what you would recommend for the best way for members of the community to get involved, to just start doing it themselves?
Yeah. And you can contact the research team using email addresses, or if you'll do that, I'll give you my mobile telephone and we can talk about it. There'll be a learning process, but I can tell you, once you get a feel for the way these things look in moist chamber, it'll be easy. You'll have a lot of fun doing this.
Yeah, I think once you see how it looks the first time, you'll know what to look out for over time.
Thanks. Okay, we have a hand raised. Sam Kieschnick, I believe, has a question. Sam?
Hi,
Hello.
Sam.
Hello, can you hear me all right?
Yes.
Good, I hope so. Really interesting project. I'm really excited about this. And I'm curious, you used living tree bark. Have you ever done it with like a fallen limb or with a bark that's in different levels of decay to see if that maybe has different cultures that pop up?
I personally haven't, but I know that people have used moist chamber cultures for a lot of different materials. So not even just wood, but even leaves as well. Brooke, I think we just learned one of our old interns, or one of our digitization technicians, Kelly Carroll, is going to be putting blades of grass in a moist chamber culture to see what fungus could grow from that. So I'm sure that you could. Again, I encourage you to try whatever you think could be interesting. We just don't know, so until we try.
Sam, let me answer that. Boy, you hit the nail on the head. The Corticulus myxomycetes that only occur on the bark surface of living trees have a special life cycle, and they're adapted to that, and they're found nowhere else. The things on dead limbs or a standing dead tree or a tree that's died where the bark is separated are species usually found on ground sites, on decayed logs. They take longer to develop. So there is a biological succession here, depending on the state of the tree, whether it's alive and healthy and how big and how old it is, versus a tree that has died. And we do know that there are ground species and corticulous species found on the bark of living trees and in the canopy. And the difference is due to, in part, their life cycle and the different stages. So that was an excellent question. You hit the nail on the head, and you're right. We're involved with succession here. And note, there's some of these things that appear early in 24 hours. Some appear later. So there's a succession of time on the tree bark. So the ecology of these things are really interesting.
Thanks, Harold. Tiana, you had a question. Would you like to ask yourself, or I can read it out? She may be busy. I'll go ahead and read it. So, question from Tiana, who's looking forward to the video of the moist chamber process. She says she'd like to do this with her children. Have you come across any resources about myxomycetes that are geared toward kids?
I like that. I mean, I really do think that children could do this and learn from this. It's really simple to do, but I haven't found really anything geared specifically towards kids. But making this video was hopefully, our contribution of, making it simple and easy to understand terms for all ages. So I hope that we can maybe start that.
Yes, I believe young children can learn this. We had a special project at the University of Central Missouri working with 7th grade Warrensburg middle school students. And I tell you, these kids found all kinds of things. Ashley hasn't mentioned all the different life forms you can see in these cultures. And with a little background information, just to give them some idea of what they look like, they find all kinds of things. And I was shocked at the diversity. And in this class, we found several new records, some for the new record for the USA, some for Missouri. And that's what you can expect to find. You're going to be surprised. You're going to be surprised.
Yes, I think that you'll be surprised. And I was even surprised to learn that a few scientists decided, they suggested that it's more so the microenvironment that determines the species distribution. So these are found worldwide. I mean, they're everywhere.
The last photograph she showed is a spiny spored species of Lycia. There are over 75 species, not a single one has spines. And this thing has a transparent peridium. There's never been a myxomycete described anywhere in the world like this. And we found it on a living tree in a nature park, Oliver Nature Park. Now, you can do the same thing. Go out there. For example, another tree that we found some interesting things on is bur oak, Quercus macrocarpa. Well, I've never even tested that tree for myxomycetes and moist chamber cultures. So use your own imagination, go out in your backyard, find a big live tree and set up your moist chambers.
Thank you, Harold. We have a good question, an interesting question from Sonia in the chat, who asks, have you tried pecan trees? Which I think is a great question, because I think pecans are known for having kind of root exudates that kill off other, highly competitive.
I haven't personally. Harold, what about you?
Yep, Bob Buchanan's there. He can tell you about the pecan trees in the Botanic Garden and a couple on the Britt campus. They have dendrothelia on them. But here's another tree. We've never tested that with moist chamber cultures. We have no idea what kind of myxomycetes occur there. And I guarantee you, you're going to find some different ones. Bob, do you want to say anything? Can you hear me?
Yeah, I can hear you now. Yeah, they're very interesting trees. They have not only very interesting things on the bark, they have very interesting things on the leaves that eat the leaves. Very interesting. I've been doing quite a bit of studies with those right now. But the bark would be very interesting to do this study with. Very, very interesting. Yeah.
Tell them what they are. They're insect galls. Tell them about the galls.
Which ones?
Well, on bur oak or on the leaves of trees.
Most of those are wasps. The little wasps that are about 1.5 millimeter long. They can't sting you, but they are very common. I've discovered 27 species of those just right here in Fort Worth. And it's just amazing how diverse they are, and they're all over the place. But you can't see them unless you really know what you're looking for.
Thanks, Bob. We have a couple questions in the chat. I want to make sure we get to them. These are questions more about the specifics of the technique. So DG McSweeney asks, any issues with leachate from the bark reacting with the metal pie plates? Maybe asking, would plastic be less reactive? And Dawn has asked for more specifics on the moist chamber.
I guess I'll ask Dawn what specifics maybe they're thinking about, but I don't know about any of those issues, but I also haven't tried doing this in any other kind of container. But you don't have, I mean, you really don't have to use a pie tin if you want. I think anything with, you know, maybe a few inches of depth and something that ability to close it in some way, you would still get that same effect.
Well, I don't think you have to worry about that. We use plastic petri dishes. We used to use in the old days glass ones. Now we use plastic and we put filter paper on the bottom. That, I don't think that's a problem. Don't worry about that. Remember, we try to keep this simple. This isn't the way we do our moist chamber bark cultures when we're trying to control all the variables. And Ashley is ideal to give this presentation because she's learning how to do this. She's just starting out. She's an amateur, and I hope she piques your curiosity so you'll start doing this.
So what I wanted to ask was, the number one, I found it curious that you put the water in after you put in the bark and the paper towels. From my way of thinking, I would have put in the paper towels and then, whoops, my husband's at the door. Sorry, gotta go. But anyway, I would have put in the water on the paper towel, then put in the bark. I mean, is that specific? Do you have to do it that way?
I don't see why that would be a bad way to do it, especially if you're trying to avoid pouring it directly up. I don't know, Harold, have you done it that way or have you always?
No, I haven't done it that way, but there's nothing wrong with that. Use your own creativity. But I recommend you don't pour a lot of water on top of the bark. Let it gradually absorb. And that can be done in different ways. It is important that you don't overlap the bark. You try to keep them spaced out so that, and by the way, when you look at the bark, they sometimes develop on the underside. So you want to take the bark with a forceps and turn it over and look on the underside, because you may see in the plasmodial stage, you may see things developing there. So it occurs on both sides, believe it or not.
Thanks. A couple more questions. Mary asks, so how do you want specimens for the herbarium prepared? I guess this would be, and this kind of relates to a question from Morgan. So you're growing these things in your moist chamber culture. Can you sample from them to make a voucher? And also, have you isolated any of these fungi for DNA sequencing?
Good questions. So again, Harold just told me how to do this the other day. Once I found a certain species that he could, you know, ID to a T. So all I had to do was take that part of the bark out of the moist chamber culture and let it slowly dry. And then put it in just a container like this. And I just put the ID on the top for now. So I could potentially, deposit this in the herbarium if I wanted to. What you can also do is create a slide, either one where you can shake out the spores on the slide or one of just the fruiting body itself. So that would be another way you could deposit in the herbarium. Right, Harold?
Let me also say that Ashley got very, very lucky. She had 4 species of Corticulus myxomycetes in a small area. You're going to find in some cases, you'll have a mixed bag of different species. Don't try to separate them. Just put them all in the same box and somehow get it to us, and we'll give the identifications and we'll log them into the herbarium. You can really help us with that.
What about extraction for DNA sequencing? Anything on that?
Nothing on my end, but Harold may have something to add.
We aren't concentrating on that right now. It's a long story and complicated. The DNA sequencing for Myxomycetes still is in its infancy. We have technical problems. It is being done, but that's something that we're not. We've got so much on our plate and table we have to publish and gather data on. We can't add that to what we're doing. And by the way, Billy Stone is preparing our scanning electron microscope stubs and doing that observation. And our photographs are the first of any kind for some of these Myxomycete species. And he just got the first SEMs ever taken for dendrothelium spores. So we're coming up with cutting edge research using the SEM in the Sumner laboratory.
Can you, so you may not be able to do DNA sequencing, but can't you test like the composition of Myxomycetes, or is that just fungi?
No, that requires a spectrophotometer. You're getting into some very sophisticated techniques, and that's beyond what we're talking about here. There are so many discoveries we can make using this technique and involving people with amateurs with beginning backgrounds that let's just concentrate on that and not try to do some of these newer sophisticated techniques. We got enough to do just doing that.
Thank you,
She's
Harold.
laughing. We have a lot on our plate.
Right, we have a good question here in the chat from Nanook. If bark is dead tissue, what difference does a live or dead tree have on the species? It's a good question.
To answer that.
Yeah, go for it, Harold.
Okay, a lot. And Sam hit that. Live trees have a certain group of Myxomycetes that grow and develop there and only there. I coined the term corticulous Myxomycetes for them. They're found nowhere else, only on the bark of living trees of different species. There are some species that occur only on leaves, on decaying leaves on ground sites. And many and most of the species occurred on dead decaying logs that are on the forest floor. And so you have these different groups of Myxomycetes that have adapted to different types of habitats. And there's even more. We just published one that occurs in at the edge of melting snowbanks in the Villa Caldera National Preserve, Arceria versicolor. We just published. So they're Myxomycetes that are specially adapted to different habitats, some in deserts. Been a lot of new species described in different deserts of the world, and you wouldn't think a desert with the desert plants would be a good place for Myxomycetes, but they are.
And I'll just add that it could also be, you know, stuff that we can't see either because the life cycle is so short, it's hard to view this out in nature. But there could be certain prey that's on live trees versus dead trees, and that could also be the reason why they're in those places instead, maybe.
There's one thing she didn't mention. A lot of these tiny lycias and echinostelias, they occur in the crevices. And as your plate dries out, these crevices or fissures stay wetter longer, and a lot of lycias develop there over time. And so that's why you have to observe these cultures for 2 or 3 weeks and let those areas dry out, and then look in those fissures to see if you can find some more. And very often you'll find a whole bunch of them, maybe 100 sporangia in a small area. And that's why it's wetter longer, and they have more time to go ahead and develop all these different sporangia. That's just another wrinkle here. I tell you, folks, this is exciting stuff. You'll have a lot of fun doing this.
Well,
I will agree. Yep.
thank you so much, Ashley and Harold, for bringing this topic to us. I know we've got some people who are really excited to go out and give it a try. And I guess this is a great time since it's really wet, and maybe we'll have an abundance of things out there. We are at the time today. Our speakers, if you'd like to add any parting wisdom, you can go ahead and do that now.
I was just going to say that I think this is really exciting because, you know, you don't have to go to the jungle to find these new species. You can really try this on your trees in your backyard and see what will grow. So you don't really have to go far. For those who aren't into like hiking up in the mountains or anything, you can walk right out your back door. And that's all I'll say. It's you know, under the microscope, it's a whole new world. And I've seriously, you know, I could only share so much for my presentation, but I've seen a lot of cool things just in the past week.
Let me say, I really enjoy the teamwork and working with a lot of different people of all backgrounds. Let me tell you, you don't have to have a PhD to do this stuff. And the camaraderie of working with a lot of my students, all the amateurs that we work with, people at Brit, that's what's fun. The teamwork that goes into this, and everybody has their own expertise. And I tell you, you're going to find things that we have never found. You'll surprise us.
Well, thank you all. I think Ashley's going to end on sharing that slide again that has their contact information. You can see that there. And don't forget to join us again next month for our lunchtime lecture, first Tuesdays every month. Thank you so much to our speakers. Have a good day, everybody.
Bye.